And I will just say that we are, there's just so many. I think there was 3 papers that came out this week that I probably could have included in this presentation. So what I decided to focus on more was instead things that are probably available to you right now in general research institutions that you could collaborate with or probably find nearby. So here's a brief outline, but I'll just jump right into some of the techniques. One interesting thing that's been happening recently is the digital analysis of well-known technology. So this is something that's been done in digital droplet PCR for a while, and the idea is that you partition your reaction, or maybe you're looking for antigens or some fluorescent marker, and you partition your reaction or your solution of EVs into a lot of different microwells. We're already applying that quite widely for digital ELISA. And the idea is that you can bypass the need for having a standard curve that you compare against. And so this is really, the details come out in the math, but briefly what this allows you to do is really fine detection of a small number of molecules when you have applications that need high accuracy and very good sensitivity. And the idea is just splitting EVs into different wells. Or droplets, and I'll show a couple examples of that. And this is being matched by the need for high sensitivity detectors to actually take advantage of that sort of technology. You can see with these type of digital applications, the flow cytometry experiments that we've heard about, this is the work from Dan Chu, who recently presented a lot of this work at our ISEV Tech conference that many of you probably were at. And you can start to see agreement with the sizes of these compared to cryo-TEM, and actually that there's a very small copy number of particular antigens on single EVs. You've heard a little bit about resistive pulse sensing, which is a technique where you have a nanoconstriction. Particles move through that. They affect the electrical properties of that constriction. If you can measure the magnitude of the signal change during a particle translocation across this channel and the time it takes to pass, you can actually extract a particle concentration and size pretty accurately from solution. There's a recent advancement to this technique, which is just recently commercially available by Spectrodyne. They're playing a major role as a sponsor in this conference, and you'll be able to find this instrument out and even test it. And they've added the ability to do fluorescence at the constriction. So now you're combining this electrical measurement, which already has a pretty low sensitivity of down to around 65 nanometer, so beating conventional diffraction limit of optical techniques like flow cytometry and NTA. But now you can measure some fluorescence off that too. So that's a very interesting technique that we're interested to try out as well. And the promise is then this overlaid information where you can get the size, histogram, as well as the sizes of those ones which are expressing a certain fluorophore. In this case, it was GFP positive EVs. This technique only requires a few microliters, and there are disposable cartridges. That's an advantage because they can be very easy to clean up, but it's also an expensive consumable. Another interesting emerging technology is super resolution microscopy. So you might have heard about this in the context of the Nobel Prize in the last decade. And this is the ability to actually beat the diffraction limit of fluorescence microscopy and has widely been applied to cellular imaging. So you can see here a conventional fluorescence image on the left and a super resolution on the right. You notice a great increase in resolution, spatial resolution. This essentially beats this problem known as the diffraction limit, where when two fluorophores are spaced closer together than the limit of resolution using about half the wavelength of the light that you're interrogating with, you can't resolve them. Instead, you use special dyes which blink on and off. So at any one point, there's just one dye in any physical spot. And then you use a Gaussian fit to localize the center of that brightness. And using this combination of localization and blinking dyes, you can actually guess where the dye is more accurately than what you can actually image with conventional diffraction limited lasers. There is a company that has commercialized this technology specifically for the application of EVs, and they're called ONI, and their first instrument is the Nano Imager. They're also here, and we'd be happy to talk to you about this. And here you can see kind of the most optimized images, which are that scale bar down there is 100 nanometers. So you can start to really pick up small copy numbers of individual or small groups of antigens. Now that's one flavor of super resolution. There's many others. One is turf microscopy, which is another specialized example where you can't get down to the super resolution of what I'm showing on the right, but there's been a lot of work in examining EV fusion and EV characterization with the slightly better resolution than you typically can. So super resolution can beat that diffraction limit. In ideal cases, you can get down to even a spatial resolution of 20 nanometer. But the models to convert this raw data into particle measurements are not that straightforward and still under development. And you require these specialized blinking dyes. And most of our non-specific dyes, like CFSE or other dyes that you might use, don't have the ability to blink very efficiently. And so there's a dye selection issue. But at least for antibody coupling, the dyes are very robust. Another interesting emerging technology is correlated light and electron microscopy. So we just heard a lot about electron microscopy. One of the weaknesses is you don't get any specific molecular information. You can do immunogold labeling. It's very challenging, expensive. There's non-specific binding problems. It takes a lot of expertise to do that. And even then, you have to crowd these giant, bulky gold nanoparticles onto EVs. The promise of this correlated sample is that you actually prepare a sample like you would for electron microscopy, but then you perform light imaging on that and you correlate that on a pixel-to-pixel scale. This has been used to study EVs deposited on surfaces down to single EV resolution, to look at shedding events and also uptake events. And so really you're combining that labeling power, that molecularly specific information of fluorescence imaging with that high resolution, high spatial resolution of electron microscopy. The problem is you get a lot of artifacts when you do this EM processing. So as we just heard about, the shrinking and drying, this is very practically challenging to do. It requires high microscopy skills and a lot of training. And you could hardly call this low throughput or high throughput. It's very slow. There's been many advances in single particle analysis, and I would say the common theme to a lot of these are really trying to multiplex protein abundance with multiple markers onto single EVs. And so with normal fluorescence imaging, you're limited to 4, 5 dyes if you're crazy. Above that requires a whole suite of experiments to detangle those and spectral unmixing and lots more advanced techniques. And so there's a couple different ways that people have tried to get around this in a different way. And a lot of this work is in the group of Ralph Weisslieder and Hako Liat in Harvard. And on the right, you see their single EV analysis technique, where they basically bind EVs down to a surface. They target them, they tag them with different fluorophore antibodies, and then they remove those and do a second batch, and remove those and do a third batch. And this way they call this sort of cyclic imaging. On the left, they have adopted the kind of drop-seq technology where you take barcoded beads and mix them in with single EVs and droplets. You have barcoded antibodies. You link those two together. You do sequencing later, and you can figure out on a single EV in a single droplet what is the copy number of a multitude of barcoded DNAs or RNAs. They're using DNA as the barcode. And this has also been further developed as a digital technique as well. And what this group has concluded with these two platforms is something quite provocative, where they claim that this conventional picture that we have of EVs that you've seen many times today as being chock full of every marker that you can find in a general proteomics run is actually not true, and that most EVs are completely scarce, mostly empty, and have a small copy number of most of the markers that we're convinced are there. I think, cautiously and conservatively, the real answer is probably somewhere in between these two pictures. The last group of techniques I want to tell you about are based on plasmonics. And these are interesting light matter effects whereby light interacts with nanostructured metal materials like gold. This is a particular type of technique featuring plasmonics called surface plasmon resonance imaging. And in this case, this is a widely commercially available instrument typically by the company Biocor. You could probably find one at your university. You can array antibodies onto the surface. Light reflects off the bottom of this thin sensor and gets absorbed only at a particular resonance angle. It sets up a standing wave at the surface. If anything binds to that region above the chip and changes the refractive index, like EVs binding to antibodies, the angle of resonance actually changes. So in a typical experiment, you can flow over using simple microfluidic processes, EVs over to antibody-coated chips, You track that change in resonance angle as you're increasingly absorbing particles onto your surface. You track that over time, and you can extract really detailed chemical information. And this has been widely applied to get a pretty low concentration detection limit for EVs based on aptamer capture. You could further couple gold nanoparticles with a secondary antibody on the other side, sort of like a sandwich assay, and get very sensitive measurements. And some people have already turned this into kind of a microfluidic chip with multiple pattern spots to look at EVs in that way. Finally, there's Raman spectroscopy, which is what my group focuses on. And this is a technique whereby some of the light that interacts with particles actually loses or gains a little bit of energy. Most of the light we're thinking about interacting with particles is totally scattered and changes, it does not change its energy. This is Rayleigh scattering. This is what we use for flow cytometry and NTA and all these other techniques. 1 in 10 million of those photons actually change energy when they interact with the EV, and they change energy by a very specific amount that's related to the vibrational or rotational modes of the molecules comprising that. We've used this extensively to spectrally analyze EVs deposited on the surface or in suspension. So this really can be a single particle technique. You see at the bottom left here, we have a single particle trapped in the laser, and we can hold it there for a few minutes and collect a Raman scattered light. In a more advanced version of this technique, we've actually been able to track the Rayleigh scattering in addition to the Raman scattering. And so as you build up multiple particles in your trap, you can track the Rayleigh scattering. You can actually use that Rayleigh scattering just like we do in flow with additional calibration to generate EV sizes. So at the same time you size, you're also collecting some of that inelastically scattered light to build spectra and tell what the particles are. This has been applied for many years at the single or bulk scale, and we've learned that, for example, tumor EVs have a higher cholesterol to lipid ratio. That is something very generically, very widely and broadly applicable to EVs. Here's the example on the right of doing sizing and spectroscopy at the same time. And the most advanced form of this recently is to control or overcome that weakness of the 1 in 30 million photons and that weak nature and having to take minutes-long measurement. Actually, if you put these particles similar to the SPR in the near field, so a few nanometer away from nanostructured gold particles, you get a huge boost in sensitivity. The Raman scattering appears 10 to the 5, maybe in some cases 10 to the 12 times higher. So you can really start doing extremely fast imaging, approaching or even surpassing conventional fluorescence imaging. And most of the recent work here has been in making lithographically ordered substrates to really get up to that higher level of sensitivity. I threw this in on the right based on the conversation this morning. This has been the primary way that you can actually distinguish lipoprotein contamination from EVs because they're spectrally different. And the most advanced formulations of this technique really have been in the combination of machine learning with this rich spectral information. And we're making great headway in this field for really advanced cancer diagnostics in very small volumes of materials using this technique. Thinking about machine learning, I'll close with that. I asked ChatGPT, what is the most exciting recent development in the characterization of extracellular vesicles? It did a pretty bad job. Morphological characterization, quantitative analysis, and cargo profiling, that's generally true. And I think we're way ahead of that if you want to be on the forefront. So if you don't want AI to come take your EV job, I think you can focus on the novel developments and you'll be just fine. So new technologies, this is a small slice. This is not even really getting into newer things that you're going to see in these high-impact modern literature using microfluidics and new adaptations. But these are ones that you probably have access to and can test on your own. And because they're new, there's many opportunities to actually inject rigor and reproducibility that you've been hearing all day into these because it just hasn't been done yet for most of these techniques. Thanks very much.